mitosis, and you will see that the centurons and two centurons make up one centrosomes, and also the spindle fiber that's all made from cytoskeletons. So it plays in very important roles in the cell divisions. And in the lesson today, we study another type of the cell division, that is the meiosis. Meiosis, the mitosis is in the normal cells, and the meiosis happens in the sick cells. What is? So this is the mitosis, and remember for me, PMET. That's 4 main stage in the mitosis. PMET, prometaphase, metaphase, anaphase, and telophase, and also the action of the chromosome in this process. And for the meiosis, or we call them the sex cell division. So let's see what are occurring during the cell meiosis. So in the first step, or when the meiosis happens, we can see that DNA strands becomes condensed to form a chromosome that we can observe the chromosome under the microscope, and they are homologous. It means that the chromosomes that are identical is one from mothers, one from fathers. They are identical. We call them the homologous, or they just look like the same, and they begin to approach each other. And in the second step here, so homologous chromosome pairs to form a bivalence, and the centurons divide and more to opposite pole of the cells. Here is the centurons in the first step. And in the second step, the centurons divide into two centurons. And also, this is one centrosome with two centurons, and they become double here. We have a 2 1 and 2. We have two centurons in each centrosomes, and they move into the opposite poles of the cells. Here I see this one, or I think it's new words, aster here. We have an aster. Aster is a cellular structure shaped like a star. It means a star. So the consisting of the centrosomes and is associated with microtubular during the early stages of the mitosis in animal cells. Aster is mean centrosomes plus fiber. Okay. The green one here is centrosomes, and you can see the number of the fibers touched with them. So we call this structure is the aster. So in the next step, the step number 3 here, the bivalence duplicates to form a tetras, or 4 chromatase group. Tetras is the 4 chromatase group. So these occur during the first phase of the meiosis, 4 chromatase. We have a tetras, 4 chromatase in the first phase of meiosis. And the nucleus membranes are started to be disappears, and we have a very important process happened here is crossing over. Crossing over in Vietnamese, 交đổi chéo. Crossing over occurs. So like the preparation for the meiosis. And the meiosis, we have two process. That's two times of mitosis is more or less the same. Like metaphase ones in the tetras, the tetras attach to the spindle fibers at the centrosomes and light up in the middle. And in the anaphase one, the tetras started to separate, and the pairs of chromatids move along the spindle to their respective centurons. So they started to separate here. And in the late anaphase ones, the chromatids are almost reach the spindle post, and the cell membrane beginning to be constrict. So they started to divide into two cells. In the telophase ones, nuclear membranes enclose the separated chromatids, and the cell membranes complete its constrictions. So in the first meiosis divisions, when it's finished, we have two cells. X cells have the same number of chromatids as the parent cells. In the prophase, this is the second phase. We have the second phase here, the number 2. This is preparations, the phase number 1 here. Meiosis 1, and we have the meiosis 2. Meiosis 2, we have another type of mitosis. So prophase, metaphase, anaphase, and telophase, we have another PMET. So in the prophase 2 begins, in the second meiotic divisions, the homologous chromatids do not duplicate, but they mirrors are separate. And in the metaphase 2, the chromatids, they are lie up in the middle. So the same, like the metaphase ones. So in the metaphase, the chromatids lie up in the middle, and the centurons and the aster at the poles and the spindle fibers started to form. And in the anaphase 2, they are now separated chromatids apart their respective poles, and the cell membrane begin to contract. And in the telophase, we have complete the meiosis divisions, and now we have 4 cells and X cell with a half number of chromatids of the parents. So it's like in when we produce the sperms, when we produce the eggs, so 4 eggs will be disappear, and only 1 egg containing all the materials from another 3 eggs. So once we produce eggs, so 1 egg is completely big like this compared to 3 others. So we have only 1 egg during the eggs formations. But the first sperm, we have the 4 equivalent sperms. Pay attention about this. And about the meiosis, no, I will note it you later. So now we see a videos about how the meiosis happen. So we see animations.
Have you ever wondered how 2 siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other, and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different, though, from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now, during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have 2 newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with 2 new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the 2 cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So it's all about the meiosis. And for the cell division in genres, just remember for me different states of the chromosomes, like what's happened to them in the metaphase, so in the meters, and what happens in the anaphase, so they separate far away from each others. So the question concerning the cell divisions, I often just in general, I just give you pictures, and you need to define that which stage of the cell divisions, like they are in the anaphase, or they are in the metaphase, or they are in the telophase, for numbers. And the second thing that you need to remember is like the number of chromosomes and also the number of chromatids. Like before interphase, we have 40 chromosomes, 40 chromatids. After the interphase, we have 40 chromosomes, but now we have 92 chromatids. Because the chromosome is multiplied into double, but they still stick in the centromere. So we count only the centromeres. We have 46 chromosomes, but we have 92 chromatids in the interphase. And after the meiosis, once we have a 22 chromosome, but 46 chromatids. And after the meiosis 2, we have a 23 chromosome and also the 23 chromatids. Just remember that's our number for me. So you can find out the numbers of chromosome and chromatids in the meiosis videos. And also remember for me, that's the recombinations process, quá trình trao đổi chéo, crossing overs or recombination process. They are contributes to the diversities in evolution. So they create genetics diversity as the level of genes and reflect differences in the DNA sequence of different organisms. So in these videos, you can see also like they show a picture about the sperms. We from one cells they can produce 4 sperm cells at the end in the end, and X4 sperm cell different from each others, and they different from the father cell also. So that's all by the recombination process or crossing over process make the gene diversity. Okay, so it's all about the mitosis and meiosis. So who can summarize for me the difference between the mitosis and the meiosis? Different between two processes. Yes, Phạm Vinh, Nguyễn Vinh Phạm, Nguyễn Phạm Vinh, Vinh Phạm, Phạm Vinh.
So as I remember, in mitosis, like
Please speak louder because
Okay.
I cannot hear you clearly.
Can you hear me? Hello.
I can hear yours, but it's too small. It's a small voice.
Okay.
Can you speak louder?
I'm speaking like pretty loud. I think it's the mic. Let me see.
Okay, keep continuous. I will try to hear. Is maybe that's a problem with my equipment.
So with mitosis is the process that the cell like makes a copy of itself, like identical copies of in body cells, and in meiosis is like mostly producing sperm cell or like for fertilizing or something, like the sperm and the egg cells, not the body cell, and they do differ from each other's like genetic material a little bit. I think it's the I think it's the thing that I can summarize.
Okay, so I think it's the big difference between them, right? One happens in the somatic cells, or you say the body cells, and one happens in the sex cells.
Yes.
One's from the mothers, or in the end, we have 2 identical cells, and they are identical to their mother cells. And about the meiosis, it happened in the sex cells. They produce just 4 daughter cells, which is different from each other and different from their mothers. They have the half of the genetics material compared to their parents. And what else? In the mitosis, we have just one. I call them this one route of the P mat, like, right?
Yes.
Prophase, metaphase, anaphase, and telophase. But in the meiosis, we have 2 rounds of them. And also pay attention about the number of chromatids and also the number of the chromosomes during the meiosis. Okay, so let's see a video about the comparison between the mitosis and the meiosis, and you can see see more information about the difference between these 2 processes.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away, the chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make 2 new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form 2 cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with 2 identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells, gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So in the videos or comparisons, the difference between the mitosis and also the meiosis. And you can see that one's words appears in nearly at the end of these videos. It's about the diploids and about the haploid. So diploids that have pairs of homologous chromosomes and haploids and nucleus that have only one chromosome of each pair. Remember, it may appear in the test. Okay, it's haploids and also the diploid. So in the videos, we can see also ones are, I think that's one image about the mitosis and also the interphase. Do you remember? Sorry. Sorry.
Yes. Yes.
Okay, there. Sorry. Apologize because my laptop had problem. So in the videos that we see, just one pictures about. You can see somewhere the interface. You see, or a circle like this, and one small piece. They show the mitosis, and the last part, the very big part, they call them interphase. Do you remember interphase in your high school?
Yes.
Or what it is? What is the interphase?
Is it the phase where the cell prepares itself for mitosis?
Prepares for the mitosis?
Like when it just lives normally and prepares itself, both of it.
Yes, they live normally and prepares. Yes, all right. Someone typing a Vietnamese word. Kỳ trung gian. Oh, sorry. Here in Vietnamese, or kỳ trung gian, we have kỳ trung gian. So let's see what happens in the interface and what happens in this circle like this. This circle we call the cell cycle. Cell cycles. And we have a different phases during these cell cycles. Here's, oh, sorry, here. So here is the cell cycles, and they last for 24 hours. Actually, it depends on the cell type. This is the cell cycles. We have the mitosis. That's we learned. That's where the cell is divisions. It's not really mitosis. Okay. This cell cycle applies in the somatic cell, so we indicate here the mitosis. And for the last things, they are interface. For the rest, very big parts, we have the interface. So you know that the center to all the biologies is the cell theories by the Sladen and Swans. And by this, we have one very famous sentence that cells arise from pre-existing cells. So in unicellular organisms, each cell division produce a completely new and independent organisms, but in large multiple organisms, thousands or more cell division begins with a single cell, so many more divisions requires over the lifetime of the animal to replace the cell loss during the courses of its life. And the cells are the cell life, we call the cell cycles here, started with the mitosis, and most of the time they live in the interface. So let's see what happened in the interface. So the interface, we have a small phase, three stage. We call them G1, S, and G2. The G1, the G here is stand for growth. G1 is grow 1, and G2 is cell grow 2, and the S is for so I would tell you before. So the G1s, tell you later. So the G1s, in the G1s, most of cells are spent in the life. So most of the cell life. So D1 is the big part of the interface here. D1 is most of the cell life. And they what happened in the G1s, because they are the cell growth, it means that they make more organelles and the protein in this stage. So they make organelles and the proteins. That's make them growth, is means make them bigger. So they spend most of lifetime in the interface, and the interface most of lifetime in this D1 stage. And the second stage of the interface, we have the S phase, and S is for the synthesis. Synthesis of DNA. So in this step, the DNA is replicates 23 pairs into 46 pairs. They are becoming double. So 23 pairs, 46 pair in this stage. So what happens in the cell growth twos? What's occurring into the Z2s? So we have growth 1 and we have the growth 2. So what difference between them? For the growth 1s, they mix organelles and the proteins, but for the G2, they are the phase that prepares for the mitosis. They prepare everything for the mitosis. It means what should they mix? They need to make the microtubule. They make microtubule. This means they make the centrosome. They make the centriones. Remember centriones? They make centrione. They make centrosome. They make microtubular. They prepare for the mitosis. They produce more proteins. But these kinds of proteins, remember, they are components of the microtubulars. They are alphas and beta tubulins. But for the G1, they are protein that is requires in the life. Another type of proteins is maybe proteins is in the components of the ribosomes, or this kinds of proteins is they are enzyme, etc. But for the G2, all the protein is just focusing on the mitosis. That's different from them. And that's why in G1s, the cell spends most of their life in the G1 in the interface, but the G2 is very short. You can see the ratio here. Longs in G1 and very short in G2. And our cells, we have, most of the cells live here in the interface. But can you tell me another type of very special cell that is not spend their lifetime in interface? This kind of cell just don't want to spend their time in the interface.
Can you repeat the question, please?
So that's I tell you that most of the cells are live here in the interface. So they spend a lot of time in the interface, most of them like this. Can you find any special or types of cells that don't want to spend a lot of time in interface?
Cancer cell?
Maybe gametes?
Gametes?
Just a guess.
So don't you think that the gametes don't want to be grows? That's just want to divide. What happened if they don't grow? Can you form the babies without growth? So cancer cell cancers just is very good ideas that the cancer cells, they don't want to grow anymore. They just want to divide. They keep divide as fast as possible. So most of cell life here is in the interface, except the cancer cells. The cancer cell, they must focus only on the cell division process. They don't want to grow anymore. So because it already takes time and takes a lot of material from the cells, they don't want to wait. And here's this only cancer. Cancer spend time the mitosis, and they shorten the cell cycles. They don't stay in the interface too long. And once once process happens is one very I think is very exciting process here, that some of the cells, they're not in the interface. They jump into one phase we call the G0s or G0, Z0s. And this means in the Z0s, they don't care about life cycles. They stay there. They work normally, and no more cell divisions here. No cell divisions. And for the G0s, I give you an example of the neuron cells. For the neuron cells, they are jumps out from the cell cycles and they stay in the G0s. And I remember that I told you before this, one cell that can divide from 40 to 60 times. So their lifetime is really lasts for several months to years. But for the neuron cells, because they jump out of the cell cycles, they are in the Z0 stage. So it means that your neuron cells is they have the age approximately to your age. Like you are 18 years old now, and your neurons is also the same age. Because they jump out from the Z1, just focusing on there is work, and they don't want to devise. So, how when the babies, babies in the first 3 years, they are devise and reaches the numbers of the neurons, and from that their neurons do not divide anymore. And also there's these two states of the Z or of the neuron cells. That's the stem cells of the neurons. It's actually the stem cells of the neurons. They're in the zero states, and we have another cell that's we call the differentiated neuron cells. Differentiated neuron cell. That is the cell is working in your brain now. So in the stem cells of the neurons in the zero state that they don't divide, but whenever we have injury with your brains, we have a stem cell, the brains will jump from Z zeros, come back to the cell cycle to healing your brains. So another types of the cell that jump into Z zeros are we have a several types like some of the heart cell, and also remember in the lesson about the mitochondria, I give you an examples of the muscle cells, multinuclear or nuclear muscle cells, like the muscle cell with numbers of nucleus inside. They are some of them jump into Z0 also. They just focus on work. They don't want to devise. So do you think that here's in each step here? They go straight forward. That's okay. The Z1 we have enough. All right, proteins and organelles. We move to the S phase. Or in S phase, the DNA just duplicates, so we move forwards to the Z2. Now, in the cells, we have several kinds of factors that make the cell considers when deciding whether or not to move forward through the cell cycles. They call these cyclins are dependence kinase and also the cyclins, cell cycle regulators. So cells are cyclin dependent kinase, a master regulator of the cell cycles, and they are active only when complexes with a cyclins protein, so they work with other. Only in the form of the CDKs plus the cyclins is become active. So what about the kinase? I remember I told you before, this kinase is when you add a phosphate group in one enzyme, is become a working stage. Like you have an ATP here, they give you donate one phosphate group here, and they become active stage. Okay, that's we call kinase. Kinase adding one phosphate group to make them work. Okay. So how we can find out the cyclins as well? The cyclin depends kinase. So scientists discovered the first cyclins when they note that the highest level of cyclins associated with onset of the mitosis in the embryo. So amounts of cyclin here, the time of fertilization, whenever mitosis happen, they found numerous of the cyclins, one protein we call the cyclins. So based on the cell cyclins, based on the cell cycles during their presence, we have formed three classes. We have G1 cyclins responsible to moving the cell through G1 to the S phase, and the S phase cyclins initiate and maintain the DNA replications. M phase or mitosis cyclin initiates the mitosis. And what about the cell cyclin depends kinase? They are the master regulator of the cell cycles, and they need to work with cyclins, right? And they are kinase. It means they require one phosphate group. And they active only with the cyclin proteins. Here in the pictures, you can see the CDK2, one kind of this, or CDK cyclin dependent kinase in these non-active. So non-active. Whenever they bind into the cyclin A's, we have a bind with cyclin. You can see the loop of them is opens and they become active, they can work. So first, I think it's important for the student come from the life science department, but for student from other majors, just remember they work with cyclins, and they are kinase. This means they need one phosphate group to work, just so. Don't need to remember think too much about this picture, okay? So what about this cyclin dependent kinase? They are very evolutionary concept. And you can find that in the yeast, there's only one type of the cyclins are complexes, just only cyclin 1, and they promote every actions of the yeast cells. But in mammalian cells, we have a number of types, several types of the cyclins. So here we have our cyclin D or cyclin D plus with the CDK4, and cyclin 2 with the cyclin CDK2 plus with the cyclin E. They are primers the actions inside the G1 staff. So for CDK4 plus cyclin D, they promote the progression from the G1 to the S phase, while CDK2 and the cyclin E, they init the replications, for etc. So just remember, for me, for the yeast cell, they are evolutionary concept. For the yeast cell, we have only one type of CDKs, but the mammalian cell, we have a number of. And for the cyclin dependent kinase, it the same like the CDK1 and cyclin B activities. They found a max level, they found the thresholds in the mitosis. So different level of the cyclins or plus CDKs, they are required to transitions. First, low levels promoting the S phase and high level promoting in the mitosis. So we have a low here and we have a high level here. This is how they work. And for this is another names are the CDK split, the cyclin complex, just it doesn't matter. Maturation promoting factor, MPF. That they found that cyclin plus CDK, cyclin plus CDK, cyclin plus CDKs is with high levels in the cell cytosol, and they call them this maturation promoting factors. So don't need to pay attention a lot in this slide. Just remember, in the mitosis, they found very high levels of the CDK and plus cyclins, and they call them the maturation promoting factor. And although the binding of cyclins is the primary means of regulating the CDK activities, other control mechanisms are also used. So they are primaries here, the kinase, the phosphorylations, they bind together, and by the help of the phosphorate, when this one phosphate group stick in here, they become active form. And when they become active, or the cell move through the Z2 in the mitosis. Another type is the proteolysis. So in the end of the mitosis, these CDK plus the cyclins are just one enzymes will come here to hydrolyze the cyclin B. They become degraded. No more cyclin B, and the structure become inactive, and now they end of the mitosis and they move into Z1s. And another types of regulations, like they can produce one type of inhibitor here. Inhibitor, they bind to this complex, and they are no more works. They become inactive. And another type is the subcellular localization. That's remember the NDS. When they export the cyclin B into the cytoplasms, the resulting in the separating of the cyclin B and the CDKs, but it's at the mitotic commitment. Phosphorylation is the key residue in the cyclin B, and they stays in the nucleus, and there is no combination between the cyclins and the CDK, so they can prevent the active of this complex. Like they cannot bind each other by separate the positions of the cyclin and position of the CDKs. So that is 4 case of the regulation regulated, but the phosphorylations are primarily mean of regulation. So it's the primary means of regulation. For the next slides, I will skip this for this year. You don't need to study about this. It's only about the anaphase chromatin complex and the cyclosterms, as when one enzymes by here, the cohesions between two or sister chromatids will separate. So you don't need to study about this. And also, for this, I told you before, some of the cells can withdraw from the cell cycle to go to the Z0 states. We have examples of the neuron cells. We have examples of some heart cell, and also the multinucleated muscle cells. So for the next, whenever the cells want to pass through their cell the state in the cell cycle to move another state, we call them, they need to pass through their checkpoint. So all the checkpoints ensure that each daughter cell receive a complete and intact copy of the genomes. When they need to move from Z1 to the S phase, they need to check if there is any DNA damage. And the S phase, the synthesis phase, the DNA multiplies into 2. They need to check also the DNA damage or incomplete replications. So only the cells have the conserved DNAs, they are complete or divide. Sorry, I have a phone call. So they have a complete replication. So from one DNA strain become 2 DNA strains without identical. It needs to complete the S phase, and if they have been tracked, it's okay. They need to move in the second step. In G2, they check again that the DNA is damaged or not. If it's okay, everything okay, the cell I have right to move into the next step, that's into the mitosis. And the mitosis, they pay attention about the number of the chromosome cells. If there are any attacks into the kinetochores, and so there's myth or there problem in the mitosis. So if there are any do we have the identical genetic materials in the daughter cell compared to the parent cells? Is all the checkpoints. So you see that all the mother checkpoint is to be sure that DNAs or genetic materials has been shaped or in the right order. Okay, so that's it, the checkpoint. Here's in the checkpoints, we have some kinds of protein that we call the guardians of the checkpoints. And we have an example here, the P53. P53, we call them the famous tumor suppressor. They are tumor suppressor. And if DNA damaged, they will trigger the expression of the P53 genes. If DNA damage, they will increase the level of P53. So what happens, what the roles of the P53? First, they will stop, they activate, and they prevent cell from entering the S phase, the cell. So it stops at the G1 stage. So it stop at the G1s, and they allow time for DNA repair to take place. And P53, the second job is they activate the DNA repairs enzymes. So first, they stop at the G1, they activate the DNA repair enzyme to repair our DNA damage. And if DNA damage is not fixable, the P53 will play its third and the final roles, they triggering the program's cell dates. So the DNA or the damaged DNA is cannot pass. So first step, first step stop, second step are calling the help from the DNA repair. And if it's not work, DNA is still damaged, they will call the third step. The final step, triggering the program cell dates. This means the cell containing the DNA damage is no more exists. They will be days. The cell with the damaged DNAs must suicide. They must kill themselves. This is the role of P53. And you can see here's the P53, or they activate the CDK inhibitor. CDK inhibitor will come to inhibit this this complex, and the cell will pause energy once. I will take a look back. So look back here. Here you can see how they can bind it. One inhibitor here. This is, for example, here P53 will call them or will make some process to curl the nucleus haze or produce some inhibitor, because I found some DNA damage here. So these kinds of inhibitor will come to bind to this or complexes, and they will stop at Z1 stage here. I will go further in the previous one, like now here's like we have a CDK. Now we have a CDK2 here in this stage. We have a CDK2 plus the cyclin E, or CDK4 plus the cyclin D. And when they have been phosphorylated, they will become working. Normally, we have a RB protein here inhibits the cells in the S phase. They inhibits. Whenever these complex CDK plus cyclins active, they will inhibit this protein. Okay, they inhibit this protein. So the synthesis can be happens. But if there is some problem with DNA damage here, DNA damage, here, we have P53 proteins. They will produce inhibitor, CDK inhibitor here. They will inhibit these two processes. So that means the RB is working normal. If nothing's inhibit the RB, they work in the normal, so they will inhibit their synthesis. This means that the cell will stop here. They stop at G1, so it takes time to repair DNA. So that's the way the P53 works. Okay, this is the end of the cell cycles and the checkpoint in the cell cycles. Discourse, okay. We will have the several minutes of the break times, and we move to the lesson number 8 right after the break time. But before we go to the break times, the IT units in our university want to try some new online teaching platform. That is the Microsoft Team. And they want to test that if all class can join into that platform. So please give me a favor that you can join into the link that I send you in the chat box to be sure to see that all the class can join or not, okay?
Hello again.
So it's okay for the Microsoft Teams. I heard from the IT unit that maybe next week we need to move from the Zooms to the Microsoft Team. Is okay with you?
Yep.
Okay, so now we move to the next lesson about the cell communications. So what about the communications? Like, in our course, I talked into you and you replies or you receive information from me. That means we are communicate. And in your daily lives also, we trust things, or we trust with each others. We sending messages through the emails, Facebook messages, everything. That means we are communicate. And it all happens with the cell. So let's see how the cell can communicate with each other. Oh, can you see my screen shares clearly?
Yes.
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 Bạn nào chưa vô được team thì cứ từ từ nhé. Mình dạy trên Zoom mà. Đây chỉ là một bài test thôi. So, for the cell communications, what about the cell communication? So, for the individual cells, like multicellular organisms, they need to sense and respond to their environments. So a free living cells or even a bacteriums must be able to track down the nutrition to tell the difference between the light and the dark, avoid the poisons, and if it's such as a cell to have its kind of social life, it must be able to communicate with other cells. I give you an example here. When the yeast cells is ready to mate, for example, it will secrete a small protein we call the mating factors. Here's how the yeast cells communicate. So, but in Saccharomyces cerevisiae, nấm nên ạ. Nhiều bạn lại cứ hỏi là phân biệt giữa tên về Saccharomyces cerevisiae cells. They are normally in the sphericals, like a round shape here, sphericals, But when they are exposed to an appropriate mating factor produced by the neighboring cells, they extend a protrusion here. You can see the protrusion here to toward the source of the factor. So this is how the yeast cells communicate with each other by secretes one type of proteins or one type of the steroids we call the mating factor. And what are multicellular cells? They are more complicated. Here in the picture, we have the pictures about the different stages of the embryos developed in the cultures. Cells the cell must interpret in the multitudes of the signal to coordinate between the behaviors. You will see that from the days one, we just have the sperm and the egg cells, it complete gamete, and they divide into 2 or 4, 8, etc. And egg cells in the embryo, they need to receive information from the other or the information inside them to define, okay, this cell, this part will become the brain, this part become the heart, and the other parts become the skin. So they need to communicate with each other, and this process is really complicated. It's not like the unicellular cells just sending some things and the neighbor can sense some things and they respond. For multicellular, it's very complicated. So let's see what are the communications in the multicellular. So in general states of the communications, we have 3 states. First, the receptions or the receptions, just like your friends or send you a message, and the first time you need to receive the information from the friend, right? We call the receptions. And the second is transaction, that we can translate what types of message they send. Okay, you think about it a little bit, and also the you need to transfer the information first from your eyes to your brain, right? And then during the analysis process, you will have the response, or you decided to reply this message, you decide to ignore him, or you decide to make him a phone call. This means the response. So we have 3 steps in the communication, and in the cell they are the same. So the first step is reception. So let's see how the cell can receive information from the other cells. So how cells detect what's going on around them and can respond in real time. Here, we have these sending cells, like your friend sending the message to you, and the target cell, that's you are. So we have the ligand, the sending cell with sendings, they are some small piece we call here this ligand. Is maybe proteins, like we need to produce the proteins, express the proteins in the form of vesicles, small vesicle like here, we call the ligands, or some free compound, or in the human fluids is whatever. So the sending cell with sendings, one complex we call the ligand. They are the molecule that's can bind to another specific molecule receptor. Here is the receptor from the target cells, and in some case delivering a signal in the process. And the ligands, we call them the signaling molecules. And what about the receptors? Receptors are the proteins molecules inside the target cells or in the surface that can receive the chemical and we call the signal. So it's not really here, it could be the receptor inside, it could be the receptor here also. And some ligand can cross through the membrane to bound to the receptor inside a cell. Is not always represent in the surface of cell membrane. So it's very specific. They are unique. Like the other cell without no receptor, they will receive no information. Like, all right, you have your phone number, so your friends or your Facebooks or mass ID, so your friends can transfer the information direct to you, but not to each other. So the cell is the same. The ligands will come to bind to receptor of the target cell. It's like the key and the lock. So the ligand can freely go to the bloodstream, but only the cell have the receptor that can bind to that ligands. It can receive the information, not the other.
Oh, I don't know. Should I play a little? I was trying to avoid you, girl. And then you.
So student, can you turn up the okay.
Yeah. 新面武蔵野藤原の春のね、やり直し。
Winson, turn off your mic.
Who's open the movies?
Teacher, you can mute him.
面白おかしく過ごさせてね。まず、
Bạn nào đang bật phim nha?
hear you.
Let me check.
Lại lại cô.
Kick him.
Let me check who is opening the films. So okay, it's turn off. Bạn nào đang xem TV thì chú ý mute hộ cô nhá. Đừng để cả lớp nghe như vậy. So, we have several types of ligands, as I show you here. We have a receptor in the surface, and we have a receptor inside cells. This can receive the information from the ligands, bind to the outside, and also the ligand can cross the cell membrane. We have two types of ligands. First, the ligand can't enter the cells. The ligand enter the cell, they must be small hydrophobics that can pass through the plasma membranes and bind to the intercellular receptor. We have examples of steroid hormones. We have a vitamin D. And also the ligand gets bind to the outside of the cells. This could be water-soluble ligand. They are polars or they are charged, so they cannot cross the plasma membrane. And some kinds of proteins, they are very large, they are very high molecular weight that they cannot pass through the membrane. So these kinds of characteristics, you know, that's the characteristic that are the compound that's cross or cannot cross this membrane. So hello, can you hear me?
Yes, we can.
Yes. And also the share screen, sorry, there's
yep.
a technical problem. So we are patient. So repeat again. We have two types of ligands that can enter the cells and bind outside the cell. So the characteristic of this ligand, you can see let's see, in the lecture about the membrane transportation, right? If they have a characteristic of the compound that can cross the cell, they could be the ligand enter the cells. And if they have the characteristic of the compound that cannot cross the cell, they are ligand bind in the outside of the cell. Let's see some kinds of example of ligands. Is made familiar with you like adrenalines. Adrenalines that's increase the blood pressure, the heart rate, and metabolism is like and cortisones affect the metabolism of proteins, and is much more familiar with you like estradiones, insulins for the diabetes, testosterone is for is male hormones like right, and this is for the females. Estradiones is so the estrogens. Estrogen is one kind of estradiol, so they induce to maintain the secondary female structure, and the testosterone is the secondary into the secondary male sexual characteristic. Or insulins, this one kind of hormones that stimulate the glucose uptake. So in the diabetes patients, right? So this is several types of very famous ligand. So we call all of them, they are ligands. So they're sending the signals like the insulins when you insert the insulins inside your bodies. They will stimulate the glucose uptake, the protein synthesis, and the lipid synthesis, etc. And also the histamines, nitric oxides, acetylcholines, etc. So in general, the hormones, they are ligands, except some are specific case like nitric oxides, they call the master cell to relaxes. And the next about the types of receptor, because we have two types of ligand, therefore we have two types of receptor, that is internal and also the external. So the internal is found in the cytoplasms of the cells. They respond to the ligands and cross the cell membrane. They have direct effects on the protein production by binding to the DNA. Here is the internal receptor. They are inside, they are already inside. Whenever the ligand come to bind, they will go directly into the DNA to generate the response, right? The second type, we have a cell surface receptor, what receive information from the ligand that cannot cross the cell membrane. They are cell surface receptor. They found on the cell membrane. They bind to ligand that do not cross the cell membrane, and they can respond in the, three kinds of response that open the channels to allow the ion to pass through the membranes. We call the ligand-gated ion channels. And they can activate an enzyme that set up a response or response inside the cells. And they can activate a proteins which is not an enzyme that can affect the cell component like the G protein Cooper or receptor. So after the ligands are binds, the receptor can respond in several ways here. They can respond in several ways, and they can allow the ion to pass through the membranes, or they activate an enzymes, or they activate the proteins with not an enzyme that can affect other cell or components. And here is you can see that the same signaling molecules may produce different results. So the first, like the same signaling, these acetylcholines, they can open the channels. So the first, they are open the channels. So in this case, they can promotion the muscle contractions. Or the seconds, when they bound to the proteins, G protein, they can affect other cell components, so they can inhibit muscle cell contraction. So it depends by the way they can bind to the receptor. So for the receive information from the environment, based on the distance between two cells, we can separate into three kinds of distance. No distance, that's two cells is close to each other. Short distance, that they are very short. And long distance, like imagine that from your head to your kidney, that is mean the very long distance. They travel through the bloodstream. So we can go to discover different types of the some communication based on the distance. The first one we call the no distance. Here are the APC antigens presenting cells. No distance, like for example, two cells. I draw here the animal cells. Two cells close with each other, standing right beside you. They also have some bridge, or we have a gap junction between them, so they are no distance. And another type of the no distance that is not, that is appears in the immune system. You know, that's immune system, our immune system, we have numbers of cell types in our system. And the immune system go to communicate with each other by no distance, and this is one example of how the no distance occur. So when you are attacked by the pathogens, or here in virus, with the MERS, the macrophage in our immune system will go to engulf, go to digest these these pathogens. For example, it's COVID-19. Okay, so it's COVID-19. So when they attack our bodies, the immune system will go to digest them, and after digest, and here this is the zoom out this. After digest the pathogens, they will have some kinds of antigens in the yellow here, and they send into their surface. We have one kind of protein major histocompatibilities complex, MHC. They send to their cell membrane. They send this yellow thing here in the cell membrane, and then the T cells, another type of the immune cells, will come to communicate with the macrophage. They communicate. Here is no distance. No distance here. So when it come to meet each other, to touch each other, it means they need to touch each other to receive information. So the T cell go to or touch the macrophage cells and to receive information from the antigen here, right? And then we activate the killer cells, come to kill all the cells affected by the COVID-19, and also they activate the B cells. B cell, we have two types. B cell will proliferation into the plasma cells. Plasma cell will produce the antibodies. Antibody will come to find the virus, and they can block the virus to macrophage will come to kill the virus more easily, or they can differentiate into the B memory cells. So in next time when you meet the COVID, again, they can differentiate into numbers of the plasma cell and to produce the antibody more efficiently and more rapidly. Okay, so the contacts between the two immune cells, we call the no distance. They need to touch each other to receive information. And what about the short distance? Short distance, here's a very famous example is about the transmit information between two neuron cells. The neuron cell, the synapse of the one neuron cells transfer information neurotransmitter to another neuron cells through the synapse here. They close together, but they do not touch to each other. We call the short distance, and the synaptics transfer the neuron transmitter between two neuron cells. We call the short distance. And what about the long distance? Long distance here. Like from our pituitary glands, they can secrete numbers of the growth hormones. And these growth hormones will come to our organs, the pituitary gland in the brain, and secrete the growth hormones. They go into the muscle, go into the bone, go into the liver, go into everywhere in your bodies to stimulate your body's growth. So pituitary glands ở đây là tuyến yên ạ. Growth hormone là có hormone tăng trưởng nếu so pay attention no. Whenever you see some products for the adolescent student, some are functional food they call the GH content, the GH here is growth or stand for the growth hormones. So they supply the GH, and because the parents expect their children will become or higher, so they supplement with the GH. So GH is growth hormones. Tuyến yên. So it means that the hormones need to travel through the bloodstream from the your brain, from the pituitary glands into your whole body. So it's very long distance. We call it long distance. For the long distance, or for the GH growth hormones, we have the balance between the growth hormones. Like when they secrete more GH, so the genetic material will activate, more exercise, more amino acids. So they use a lot of sugar, so the blood sugar will decrease. And they use also the fatty acids to contract some structure inside your body. So the fatty acids is quite low, like here. They come to the liver secretes insulins like growth factors, and they stimulate the cell divisions, protein synthesis, bond elongation, so make you grow, right? Grows. So because these kinds of produce use a lot of sugar from your cells, so it's low amino acids, and then they will feedback, right? The feedback to the your muscle, your glucose, and they have a negative feedback to your pituitary gland. So they grow too fast. We don't have enough glucose. We don't have enough amino acids. We don't have enough sugars for growing. So stop. So you need to decrease the levels of growth hormones. So if this two process is not in balance, it may resulting into too tall or too short person, right? It's too much process here, too tall or inhibits much, they cannot grow. You have a too short. So it's the three general thing of distance, but in the scientific words, we divide this into 5 cases. First, endocrines. Endocrine, they are long distance. Long distance, we call endocrines, while one cell secretes ligands, in this case hormone, and this needs to go through the bloodstream to reach the target cells, is the long distance. And the second case, paracrines. Paracrines, we can call them the local distance. Local. It's local because remember the first slide I showed you in this lecture about the sending the mating factors of the yeast cell, right? They are local. And also the synaptic. This is the short distance, or we call the local. And the synaptics is another case of the short distance also. They are quite short. They are short distance, but of a specific case of short distance. And the next about the contact dependence, they are no distant, like in a email system or email cells connect to these others. I show you no distant. We call the contact dependent. And one very specific case, we have outer screen signaling. Outer screen signaling is when one cell send information to stimulate themselves. And can you tell me, and which kind of cell, or we can call the outer screen signaling? One case of the cell that's sending the information to stimulate themselves. So it's the case of cancer. Cancer cells relieve ligands. Most of the cases, ligands stimulate them to divide. So they send ligand to themselves to make or encourage them grows more divides more rapidly. So in the cancer cell specific case, they are outer screen signaling. So what about the local distance? Can you tell me the characteristics, different ways by which it could be the cell could be remain the local stimuli? So these kinds of ligands cannot move or too far away from their areas, but they just want to sending information to the cells surrounding them only. So how they can prevent this kinds of ligand travel too far away?
I think they are hydrophobic, so they cannot
Hydrophobic.
go to the bloodstream. Yes.
Do you think that the hydrophobic compounds cannot go through the bloodstream?
I think that they don't have the protein that support them to go into bloodstreams like another hydrophobic substances that they still can go into bloodstream, but with the help of the protein, hydrophilic protein. So I think this signal, this ligand don't have the protein to help them to go in the bloodstreams. So they will just stay around the cell.
There is number of types of the number of things that go through travel throughout bloodstream. Even the hydrophilics or very large molecules, numbers of big proteins go through the bloodstream. So one thing that they want to prevent these ligands travel too far away from them. So first, or it could be unstable. So they can degrade by the extracellular enzymes. So if it's not taken up rapidly by the neighbor target cells, it could be degraded. And another thing that, that these kinds of reasons, we can call them. So they are specific reasons, and they will have the extracellular matrix, the matrix here. Extracellular matrix. So the ligand need to stop here. That's the characteristic of X area is not the same. It's not the same in our body also. And even in the first slide, when I show you the the yeast cell sending the mating factor. So the environment surrounding them is different from the from the environment far away from them. So we call it the extracellular matrix. Extracellular matrix. So the ligands first needs to be unstable. They easily degrade. And the second, they will need to start in the extracellular matrix so they can prevent these kinds of ligands travel far away from them. So to be sure to ensure that only the neighbor cell can receive information from them. So this is the first part about the receptions, how the cell can receive information from or there's neighbor cell or from other cell from far away from them. And the second step is about transductions, how they can transmit this kinds of information. So for the transductions, for example, they are a signal is a function that conveys information about the phenomenon. And the transduction is the process whereby one type of signal is converted to another. So example is when you have a mobile phones, these mobile phones is receive a radio signals and is need to convert it into the sound signals. When this transmitting the signals and this the reverse. So this process we call this transduction. So it's the same like a more the same like in the cells. We or the target cell receive information from the molecules A, and we need to process. We need a process here. We need to process of this transduction. These informations into the information that the cell can read. For the signal transductions, we have several cases. And mostly it occurs when the cell surface receptor that when the receptor inside a cell that they can bring these ligands direct into the DNA here. So it's nucleus here. Signal transduction occurs only with the cell surface receptor. So how they can occur? So first, by the phosphorylation. Phosphorylations are kinase. Remember kinase from the previous lessons? Kinase is mean that they need to put one phosphate group to another molecules, and it could be phosphorylate them and can active them, can change them from the inactive form into the active form. So here the signal in, so you can see here, signal in here, they will in the inactive, they off here. So the ATPs, the energy monies of the cell comes to donate one P group, one phosphate group here. It's from inactive into the active form. And if the phosphate group relief is become change from the active into inactive. And it's the same like it's with the GTPs, guanosine or triphosphate is the same. If we donate phosphate, it become active, and if really phosphate, it become inactive. So it's called the phosphorylation. This first time the transduction. So in signal transduction by the phosphorylation, because we have the MAP kinase signaling cascades. So this a very complicated part. So MAP kinase signaling cascades is the mitogens activated protein kinase. We call it the MAP kinase signaling cascade. Ở cái này dòng trong tiếng Việt Indian Army dòng tháp proteins kinase hoạt hóa bằng mitogen hình như là thế, là dịch cũng khá là khó hiểu. So how it works It just by remember here the growth factor here. Growth factor is ligands. They are hormone. They are ligands. They come to bind to the receptor here. The receptor will activate one type of proteins, kinase, MKK, and these after or these activates, they will donate another type. They will activate another type of proteins, kinase, MKKs, and the MKKs will be phosphorylate another type of proteins like MAF kinase, MAKs. And by the last, the MAKs here will go through the reactivase will go to the nucleus, go through the nucleus to phosphorylate other transcription factor here. These transcription factors, after they will be phosphorylate, after they receive one phosphate group here, this will be activated and will go to the genes and promote the genes for the cell growth and the division. So is PPP, so it means that we call the cascade like like a waterfall. So one thing attacks another thing attacks another things, and by the end, you can stimulate one process inside the nucleus we call the phosphorylation. It seems complicated, but it's a simply graph of the phosphorylation. Actually, the map kinase grow and differentiation is more complicated. Like you see in the slide here, it's not really one gene or one ligand can promote any growth or differentiation. They are the complexes of the ligands and the transduction inside a cell. It's too complicated. To so many pathway to or activate the ERK here. MEKs, ERKs here, and then to activate genes here, the genes, numbers of genes inside the nucleus. So children need to remember this graph. Just remember that some things phosphorylate some things, and this become the phosphorylate. This will become the activates, and this will active another things. It make a cascade. That's all. That's how the cell can transmit the information from the outside into the inside. Bạn nào lại đăng bật đi? Trần Đức Tuấn.
Không phải bạn đấy đâu, là bạn BI11249.
BI11249. Không tìm thấy nhỉ?
Trong cái cốt phần mute everyone đấy để dễ hơn.
BI11249. Bạn này cô sẽ. So BI11249 is the second time that you disturb our class. So it's the first time, okay? If this happened the second time, I will reduce 1 point from the midterm test. You can do another theory, but do not disturb our class. So this is about the MAP kinase. And another route of the signal transduction, we call this secondary messenger. For the second messengers, they are in response to the signals and enzymes we call the adenylene cyclase. We convert the ATP into the CCAMs here. Adenylene cyclase, they convert one ATP into the CAMP. And this is the second messenger. So they also have the spread a signal through the cytoplasms after bind to the receptor. They are the same. are more or less the same like the phosphorylation, but they require the second messenger, or they require adenylines. Here's when one ATP becomes T or CAMP. CAMP will activate another kind of protein kinase from the inactive into the active form, and they are attached into the cellular response. Here an examples of the adrenalines stimulates the glycogen breakdown in the skeletal muscle cells. Adrenalines here, they will activate or GPCR, that's adrenergic receptor. They activate the receptor. The receptor will activate the adenylate cyclase here. They activate another type of proteins, very important protein to the second messenger. And this, by the help of one ATP, they will convert one ATP into the cyclic AMP. This cyclic AMP will go to activate one protein we call the PAK. This is the kinase proteins. And this kinase protein, they will activate another phosphorylase kinase from inactive into the active form. In our cells, this kind of phosphorylase kinase or phosphorylase kinase will active the glycogen phosphorylase inactive into the active form to break down the glycogens. So do you think this process is very low or very quick? Do you think it quick or slow? It seems really complicated, right?
Yeah.
Obviously quick.
It must be quick. Like, you know, that one years injects the adrenalines into your veins. This kind of process is like happens immediately. Okay, so it seems very complicated, but is happened in very quick. This might be quick. So what about the response? So it's about the signal transduction, how the information the cell receive can transmit into the target or size, and then how the cell can respond in the third part, how the cell to respond we can discover in this part. The cells are first, the same signal molecules can induce different response in different target cells. So in the same, like we have the acetylcholines here, when they attach into the host pacemaker cells, they will decrease the rate of the pharynx, decrease the heart rate. But if they come to affect the salivary gland cells, this will secrete more. And if they affect in the skeletal, as will the contraction, muscle contraction will happen. So the same signal can induce different response in different target cells. How the cells responds? First, here every cell type displays a set of receptor protein that enable it to respond to a specific set of extracellular signal molecules to produce by other cells. These signal molecules work in combination together to regulate the behavior of the cells. As you can see here, one cell that needs to receive these kinds of ligands of the survivors. So if they receive A, B, and C ligand, they know that's okay, I need to survive. And if they receive more about the ligand D and E, they know that's okay, I receive a signal that I need to grow, become bigger, and also divide. And what if they receive another type of ligands, another type of signals that make them differentiate into working type of the cells. But if one cells receive nothing, so they don't receive any order from the center, from your brains, or from anywhere, they will die, they will kill themselves by the apoptosis. It's not like humans. When we receive no information from the environments, no message from your friends, from your parents, or you receive no information from the environment surrounding you, you're still alive. But the cells, if they lack of at least the ligand that stimulate them, okay, let's survive, they will be dies. If they receive nothing, they will be dies. They kill themselves by apoptosis. And here's how the cell respond to the signals. That's I give you an example is about the adrenalines. They're very fast, second to minutes. It's just because they do not enter, they just enter the protein function, but not enter the protein synthesis. So it's very fast. But if these kinds of ligands need to stimulate the gene expression, they enter the protein synthesis. It could be very slow for minute to hour. So here, quick change in the cell movement, secretions, or metabolism, but not involving the changes in the gene expression is very quick. But if the cell differentiation or increase the cell growth and division involved in the changes in the gene expression and the synthesis of new proteins is very slow. So we have a different route of the response. So yeah, some small hydrophobic hormones combine to intracellular receptor, and they act as transcription regulator. They are not identical, but they are evolutionary relate, belong to the nuclear receptor subfamilies. This is kind of, for example, the cortisones. They can go directly inside the plasma membranes. There are ligands that can go inside. They do not need to touch to the receptor in the cell membranes. So they can go and bind directly to the nuclear receptor proteins and go direct into our genes to enter the gene expression, so to make the transcription of the target genes. So this is a steroid hormone, the corticoids, activating a transcription regulator. What happened with the cryostorons? The cryostorons is the same structures like other steroid hormones. So what if the steroids is not normally found in the cell membranes? Could it be used as the effectively as the hormones? Could it be? If they not work as the components of cell membranes, could it be a hormone? They're in the same group. Now it's cortisones, we have estradiol, we have testosterone. They have the same structure like restorans. So why estradiol, testosterone become the hormones, but not cholesterol? So if cholesterol do not found in the cell membranes, could it be a hormones like the estradiol and testosterone?
I don't think so, because cholesterol is hydrophobic. It does not dissolve in water, so it cannot be a hormone.
Hydrophobic. So
Yes.
what about the OS group here? OS group is mean they are polar, and this mean they are hydrophilic.
But
They're polar here. And they have also the polar group here. Polar. It means they are hydrophilic. So why not with the cholesterol?
Because the carbon the hydrocarbon tails of the cholesterol is hydrophobic. So
So you mean this strain is hydrophobic?
yes.
So what about the polar group here? What
Mm,
difference?
well, it gets stuck in the membrane.
Assume that there is not the component of the cell membrane.
That is too long.
It's too long. The tail here is too long.
Yeah, it may get stuck in some structure along the way.
It's stuck in somewhere, right?
Yeah.
So it's okay. I agree with you that the polar group here, it just have only one polar group, but it's too hydrophobic because it's to be an effective hormone by itself. Okay, it's because it is virtually insoluble in waters and it could not move readily as a messenger from one cell to another cell, just because they are too have a very long tail here. They are too hydrophobic. Okay. All right, send me your names and your student IDs. Is maybe I give you some extra points. So for the next about the response of the cells, response of the cells could be the proteins, could be an enzyme that's produce large number of small intracellular signalings, that is cyclic AMP or cyclic GMPs, or it could be an enzyme that modifies large number of intracellular target proteins, for example, or phosphorylations. And here is many extracellular signal acts via cell surface receptor to change the behavior of the target cell, like or from one kind of signals through the or signal transductions. It affect the metabolic enzymes. It affect the cytoscatters. It affect also the transcription regulator. How in the cells can respond to this kind of signals by change in the gene expressions as ones, or increase the intracellular metabolism too. The number 2 is very quick, remember? The first one is very slow. The cell growth, they do not normally divide unless they are stimulated by signal from others, or the cell death if they receive no information from the environment. It could go to kill themselves, or first no information, or information does command them to be died. Remember the P53 that sends information to the cell that make them die because they have a damaged DNA, right? So in the case of the cell death, it could be no information received, nothing, no information events, or order them, command them to survive, no information, they were dead, or the information that's called them, okay, you have the DNA damage, so you need to suicide. This is the how the cells respond to the signals. I know this lesson is quite complicated. It's a little bit difficult for you, but try study harder. And okay, I have a videos about the cell communications, and I hope that you, after seeing these kinds of videos, you have more. It's easy for you to to learn this kind of the cells communication.
Professor, we can't hear the audio. We only see the video but no audio.
A question for you. Have you ever played the game Telephone? You know, the game where someone whispers something to someone, and then that person whispers it to someone else, and then that person whispers it to someone else. And by the time you get through everyone playing, the original message is all messed up. I used to kind of dread that game. Most people seem to like it, but somehow whenever it reached me, the message was always really messed up, so I felt like I was spreading nonsense. Well, either that or it was me all along that misheard it. Anyway, the game is all about communication and how things spread. Communication is incredibly important, not just for us, but for the things that we are made up of, like our cells. Cells make up all living things, and while they don't talk in the way that you and I do, it's important for their messages, their signaling, to be transmitted and received appropriately. Multicellular organisms need their many cells to be able to work together to carry out functions. Just consider all the cells working together in one of your organs, like your heart, for example. We're going to be talking a lot about receptors. A receptor is a molecule, such as a protein, where a signal molecule can bind. One place you can find receptors is on the surface of a cell membrane. When a signal molecule binds the receptor, amazing things can happen. The receptor might start activating another molecule for an action to happen. The receptor often changes its shape slightly in the process. More about that later. So, receptor, signal molecules. The signal molecule can be considered a ligand, a fancy term which basically means that it's the smaller molecule that binds to a typically larger molecule. Signal molecules can be a variety of things, such as gas molecules or hydrophobic biomolecules like lipids or hydrophilic biomolecules like some kinds of proteins. But ligands are generally smaller than the receptors they bind. Ligands and receptors can have a very specific fit as well. Let's talk about the general sequence of cell signaling. First, reception. Typically, a signal molecule binds a receptor. Second, transduction. The receptor gets activated by this binding. This often means the receptor will change its shape. It could even involve a whole series of molecules changing their conformation in something called a signal transduction pathway. This can amplify the original signal. Third, response. There's some kind of response that is going to happen. A portion of DNA that is found in the nucleus getting transcribed, for example, that's a type of response. Now, cell signaling can involve intracellular signaling, which occurs within the cell itself, and intercellular signaling, where a cell communicates with another cell. In many cases, signaling involves both, signaling between cells and then also signaling within cells. Now, when we're talking about signals traveling from one cell to another, distance matters. Some cells are close and have direct contact. In the case of these two animal cells, they are gap junction close. Gap junctions in these animal cells, or plasmodesmata in plant cells, are connections between two close together cells that allow ions or other small molecules to pass, and by doing so, they don't have to pass across the cell membrane. Paracrine signaling allows a cell to target another cell by a signal molecule that may diffuse between them. These cells are still close, but need not be connected. The ligands in paracrine signaling tend to be rapidly absorbed and rapidly degraded. The ligands are typically not traveling far, as this is local signaling. Synaptic signaling, which specifically involves neurotransmitters in a synapse, is another example of this local signaling. And what about long distance? Endocrine signaling can allow a cell to communicate with a target cell from far away. Signals may be carried in the bloodstream. Hormones released by certain types of endocrine cells are a great example. We also want to point out that a cell could just signal itself. For example, in autocrine signaling, a cell could secrete a certain type of molecule which then binds to its own receptor and causes a response. A cell releasing its own growth factor could be an example of this. Let's just give a few signaling examples so we can get a basic understanding of the vocab and what this can look like. Let's say we have a steroid hormone that travels through the cell's semi-permeable membrane. Remember, that would mean the steroid hormone is our signal molecule, our ligand. Once inside the cell, it binds to a protein receptor within the cell. Now, the protein receptor is active. The protein receptor travels into the nucleus where the cell's DNA is found. This protein receptor binds to DNA and is involved in getting transcription of a certain gene going, which eventually can be used to produce a specific protein. This is an example of signaling inside the cell, simplified a bit. Now, in the example, the receptor was inside the cell, a cytoplasmic receptor. But it doesn't have to be. Receptors are frequently part of the cell's membrane surface. If a receptor is sitting outside of the cell membrane surface, then the ligand doesn't have to come in. In fact, there could be properties of the ligand that may not allow it to pass through the membrane. The ligand could be hydrophilic, which would make it hard to pass through. Let's mention an example of a cell surface receptor type, a ligand-gated ion channel. Ions normally don't go unassisted through the cell membrane. They are charged after all. See our cell transport video. But a ligand-gated ion channel gives them a way through a channel. But it is controlled. In this example, the channel is closed. But here comes a signal molecule, a ligand. It binds the receptor, which is the channel protein in this case. Now, the channel protein responds by opening. The ions, not the ligand, go through. Once ions get through, the concentration of ions increases in the cell. And you may wonder, okay, so now there are ions in the cell. Why does that matter? Well, the increasing ion concentration can trigger a cellular response. After reaching a certain concentration and stimulating a cell response, the ligand can leave its binding site from the receptor, and the channel can close. Ligand-gated ion channels can be used by neurons. A neurotransmitter may be the ligand for the channel to open. This could happen at a synapse. But just be aware that not all ion channels are ligand-gated ion channels. Ion channels can be gated by other things. A voltage-gated ion channel, for example, depends on electrical membrane potential, not a ligand. By the way, voltage-gated ion channels are used by neurons too. Check out action potential in neurons to learn more. There are other types of cell membrane surface receptors that we don't have time to go into in this video. G-protein-linked receptors and enzyme-linked receptors are two other types that we encourage you to explore. So in summary, why do we care about the cell signaling thing? Well, realize that your body processes that keep you alive rely on your cell's ability of cell signaling. From the regulation of your heartbeat to the hormone signals traveling long distances in your body to the way neurons in your brain communicate, your multicellular self needs cell signaling. But there are many disorders where cell signaling does not work as it should, and so understanding all of the complexities of cell signaling is critical in order to find ways to treat them. Cancer is an example of a disease that can involve body cells with problems in cell signaling. We mentioned autocrine signaling. A cancer cell could have a problem where it produces too much of its own growth factor, causing excessive cellular division. Cancer cells can also have many other cell signaling difficulties where they do not function like normal healthy cells. Or another example, there are also pathogens, such as viruses or bacteria, that can take advantage of cell signaling. Consider the virus HIV, which targets helper T cells. Helper T cells are important immune cells in your body, and they have something called a CD4 receptor on their surface. That receptor is so important so that helper T cells can communicate with other immune cells. But as mentioned in our viruses video, HIV targets that CD4 receptor. It is because of the virus binding to that receptor on helper T cells that the virus can attach and infect the cell in the first place, a fact that continues to be researched for treatment options. The understanding of the details of cell signaling continues to expand. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So it's the end of the lesson today about the cell communications. I will send you a attendance checks in the chat box, okay?